Okay, well, good morning, everyone. Thanks for coming and joining us for our last Plant Breeding Center seminar of the year. I think we have a great set of speakers this last year, so we'll resume next year. But for now, this is the last one, and it's very much our pleasure to have Fikret Isik here with us. Fikret is a professor at the Department of Forestry and Environmental Resources at North Carolina State University, and he did all of his degrees in Turkey. He did his PhD in Akdeniz University in Turkey, which I just learned means Mediterranean in Turkish. And his research program at North Carolina State University focuses on quantitative genetics for disease resistance and for breeding methodology in pinus species, specifically the hard pines you work in.
Soft pines.
Soft pines. Okay, he works in the soft pines at North Carolina State University. And he also teaches classes in advanced topics in quantitative genetics and breeding, as well as quantitative forest genetics. And like I said, his research really aims at fundamentally changing forestry breeding using genomics. And I also want to mention that he's a co-author of a book entitled Genetic Data Analysis for Animal and Plant Breeding, which is a book that you may be familiar with because I used some of that book in my class, and it's a really terrific book, and it's been a big help for my research as well as my teaching. So if you don't know that book, check it out. It has a lot of useful R scripts for using ASRM and things like that, and applications of mixed models to multi-environment trials and genomic selection and so forth. So without further ado, I'll turn it over to Dr. Isik.
Thank you, Aaron, for the introduction. Good morning, everybody. It's really a pleasure to be here. My first time in Minnesota. Mentioning about the book, we have been working on the second edition of the book, switching completely to our scripts, relying less on some packages. So, packages can go away. So hopefully it will come out. It's very difficult to give a time, but hopefully next year. So this picture shows pine plantations in the southern United States. That starts from Virginia all the way to Texas, including Tennessee. It's a very large area. They do a lot of pine farming in the region. And do you know which country produces more wood than any other country in the world? Any guess? Canada. Russia, China.
Really?
Southern United States is number one. It's the wood basket of the world. So the region produces about 19% of the wood supply in the world. So it's a big industry. So my research program actually supports that industry in the South. So yes, pine plantations, I think there are around 30 million acres of area planted with pine, mostly loblolly pine. And it doesn't have major problems except that, fusiform rust disease. It is caused by an endemic pathogen, Coronium quercum fusiforme. The pathogen has co-evolved with pine over millions of years. So we believe there is an arms race going between the fungus and the southern pines. So it infects mostly young pine trees. After they are planted in a couple of years, they get infected. It's about 10% infection rate we see. And then it develops those galls. And usually it kills the trees. If not, trees become bush and not useful. So it's a big economic loss for landowners. So the fungus is endemic to the southern United States. And it's a biotropic, microcyclic, and heterogeneous fungus. It alternates between oaks. It infects multiple oaks, and then switch to the pine, infects pine. But it really damages the pine developing those galls. It uses oaks just to finish the breeding cycle, the life cycle. So research on discovering genes in pine for disease resistance dates back to 1995, maybe a little bit earlier. A group at Penn State University, led by Ronald Sederov, they discovered the first major resistant gene in one of the pedigrees. And in that research, they used the single spore isolates to challenge the pine QTL. And they used about like 300 rapid markers. Those are, they are not around anymore. So using the same method, the group discovered additional 8 QTLs in different pedigrees. Again, challenging the host genes with a single spore isolates, specific single spore isolates. So based on that research, they developed that theory. The pathogen genes and pine genes, when pine is infected with single spore isolates, we see Mendelian one-to-one segregation in the progeny. So half of the seedlings got galls, half doesn't. So it's a classical Mendelian inheritance. That's what they proposed based on those studies. So I later worked with that group, analyzing some artificial inoculation data. In that research, we used a mixed pathogen inocular instead of using single spore isolates of the fungus. So it's a bulk, spores used, sampled across the region, using a high density, we challenge those FR genes in different pedigrees. So this shows how complex actually the resistance is in the pine pedigree, in the pine populations. What you see here is that the genotype, like the resistant allele, susceptible allele in different pedigrees, there are 8 of them. And this is one of the QTL, another one. And then in the columns, you see different pathogen populations. Those are mixed spores, not single spore isolates. So the recessive alleles, like a little r1 or little r2, they were universally susceptible. The progeny having those recessive alleles, they got disease, 70–80% across different inocula, very predictable. But resistant alleles, like a big R1, big R2, they interacted with with pathogen populations. So they were more like stable. Sometimes they were very resistant. Sometimes they get infection like around 30%, depends on the pathogen. So I think the message we got from that research, the genetic base of disease resistance in pine at the population level is more complex. So yes, single spore isolates, one QTL, it follows the Mendelian one-to-one segregation, but at the population level, it behaves like almost polygenic because you have a lot of QTLs in different pedigrees, and so it's more complex. So with funding from USDA and NIFA, I received 4 or 5 years ago. We decided to look at some research questions about the genetic base of resistance in the host species. So one of the questions we want to answer is, are there large effect broad spectrum QTLs conferring resistance in the host? Or are they all like a single QTL conferring resistance to specific fungal isolates? Another question, are those QTLs effective in real forest environment? in those previous studies I just mentioned, they really didn't validate those QTLs in a real forest environment, in a plantation environment. They were all greenhouse artificial inoculation studies, right? And so if there are expressed genes associated with those QTLs, are they similar to other plant resistant genes? Those are analog type of genes we see in plants, very common in plant species. So the research led by my former PhD student, Eddie Lauer. So there are some graduate students, they really make you look good. And Eddie was one of those rare graduate students, really smart. And now he works for Illinois seed company in Wisconsin as a corn breeder. So for the QTL discovery, we use two different pedigrees. One is that cross, we call it, cross E4. The paternal parent 4, we believe had, some disease QTLs, resistant QTLs. So this shows like a breeding values of the parents. This of the 32%. The resistant parent has like 3% based on those field trials. Because they get infected in progeny test, we get breeding values of those parents. And that's the cross we selected. So we got about more than 1,000 positive progeny from that cross. And then the other pedigree is a parent 9. Again, it has, a good breeding value, much less, disease compared to sets of parents. So this cross has about 920 seedlings, and they were labeled, and we collected gametophytes from those progeny for DNA extraction. So when the seedlings were about 3 months old, they were we used artificial inoculation to get the disease scoring. And the average disease incidence for the first family was about 28%. So that suggests maybe there are 2 QTLs conferring resistance. And the other cross, it was about 54%. Again, following a single gene segregating in the progeny. So for that inoculation, we used a very dense inocula, about 100,000 spores per milliliter, sampled across the region. So it's very diverse mixed inocula. That's the fungal population we used. So, we used megagametophyte tissue from those progeny. Megagametophyte is a haplotype tissue in conifers. It gets inherited from the maternal parent. So we genotyped those seedlings, those seed progeny, which was using that seed array we developed for pine, loblolly pine, 2018, I think it was published. So after 7 months post-inoculation, we scored the disease. It shows like a small gall in the stem, or you don't see galls. It's like yes or no type of trace. So we use the two-way pseudo test cross design to produce linkage net for two different families. And why two-way pseudo test cross? Because pine is a heterozygous diploid species. And so the markers we used, they were in back cross configuration, AD, BB, or BB and B. So when you use that configuration, you get two separate maps for maternal and paternal parents. And then you can use inter-cross configuration of the markers to produce a sex averaged linkage map for each chromosome. I'm sure it's quite different from cell species or I don't know, polyploids, but much. So this shows genome-wide LOD profile for two families. The red one is the first pedigree cross E4, and the second one is the cross E9. And there are actually two loci on the paternal linkage group 2 for that cross coming from the parent, and then one locus on paternal linkage group 7. You have like a chromosome for paternal methyl parents. So when Eddie used the multi-QTL approach to analyze the data, of course, it improved, and what you see in the inset, actually two QTLs segregating in that family. So the model was improved, and it clearly shows there are actually two QTLs on that linkage group 2. So these two QTLs in parent 4, they together explain 31% of the variation in the disease incidence. And the other QTL in other pedigree, it explains only 14%. So this shows the confidence intervals of the QTLs here on the linkage group 2. Grid 1, we named the grid 1 and grid 2. They are separated about 30 centimorgans. And both QTLs were linked with a number of synapses based on GWAS. Those are synapses you see in the blue colors. They are actually tightly linked to those two loci. So, so this graphic shows the two locus haplotype effects. So for two genes, you get 4 different haplotypes. Those are the double septal haplotypes, double resistant haplotypes. And if you look at the counts for double septal and resistant haplotypes, actually shows these two QTLs are linked to repulsion on the same chromosome. The counts, those are tells you the linkage phase of those QTLs. But what was striking that the disease probability for two locus haplotypes, and when the progeny, there were about 156 progeny with two resistant alleles, the only one sibling had a gall, so out of 156. So we believe we made a mistake when we were phenotyping those progeny, because only one has a gall. Sometimes it was difficult to call the galls or no galls, and we believe we made a mistake. So our conclusion was that if the progeny inherit into resistant alleles, they are completely immune to disease. That's the probability of getting disease. It's very low. It's almost 0. Because, yeah. And then double recessive haplotypes, they got hammered, like 90%, 92%, or the probability of getting a disease like a 0.92. So that was all nice and dandy, greenhouse experiment, artificial inoculation. We got those great linkage maps, and we showed those, progeny with QTLs are highly resistant to disease. But does it really work in the real forest environment? Can we validate in a different population? Otherwise, it will be another QTL QTL study collecting dust on the shells, and there are a lot of them in forest tree genetics in tree breeding, QTL discovery, and they are not useful for different reasons. One of them is the validation; it's not there. And so, so we decided to validate those QTLs in a experiment. Those are like two, in that experiment, those are like actually genetic field tests. In that population, we used 21 parents. They were crossed using dial mating design. And so we produced about 51 crosses. Each cross has about like 45 progeny. So okay, that's the population. And those progeny were cloned, and they were tested across 8 different locations in the southern United States. So one copy of each clone planted at each location. So the test was at age 6, I think we assessed to see if those trees have droughts or no droughts. Of course, the discovery, QTL discovery population parents, those are like parent 4 and parent 9, they were actually extensively used in crosses, like 51 crosses. Those 2 pedigrees with QTLs, they were used in the dialar mating design. So there was extensive pedigree relationship between the discovery and that validation population. They were not completely genetically independent, and probably it wouldn't work if they are completely independent. Those QTLs segregated in the pedigrees in specific. So we collected data and we genotyped all the clones, 2,300 clones, with the same array, same markers. So this shows GWAS studies showing effect of the markers. So again, we detected some markers, SMP markers on linkage group 2, significantly associated with the disease, but also linkage group 7. You see probably there's like covered a little with a nest zoom. So again, we observed markers tightly linked to disease resistance into linkage groups in the same genomic regions. So that was really a great news and exciting news that those QTLs are real. They work in the real forest environment, not just in artificial inoculations. So this Q plot shows those QTLs significantly associated with the disease. You see deviation from the expectations. Large number of markers, they follow the expectations. But a few of them are, completely deviate, again showing the effect on the disease outcome. So this shows the variation for disease in the validation population. Again, we see the PC1, PC2, those are principal components. They are calculated from the loadings of the clones and using genetic relationships between those clone DNA markers. Shows the population structure, basically. So you see like 3 different groups, red, purple, and green, and then some in between, again showing 3 different dialyls. They are not completely separate because some pairs were used in multiple dialyls. But I think what's important in this graphic is that at the bottom you see the fusiform rust disease incidence. You can ignore the bottom part. So if we look at this part, you see the RAS incidence of the clones. It starts from like 0, no disease, and then all the way to, I don't know, somewhere like 80%, 90%. You see, those are the resistant clones for different dialects. And those are susceptible clones from dialect 1 and susceptible clones from another dialect, and then green is another dialect. So you see that range of disease and very resistant clones in each dialect. So the disease incidence is very independent of the population structure, because the way the mating design included resistant parents in multiple crosses in dialects. So as a complement to the QTL mapping approach, we carried out a bulk segregant analysis to see if there are candidate genes in those loci. So in that approach, before inoculation, we prepared PacBio isocyclic libraries to obtain family-specific reference transcriptomes, okay? One for each cross, one for each family. And then after the inoculation, we had the diseased and non-diseased groups, and then we sampled multiple individuals in each category. Those are 3 replications in each. And then we used Illumina libraries to sequence, and then those short reads from the Illumina sequencing, they were aligned against the PacBio reference transcriptome for each family. And then, of course, variants were called, and the read counts from the Illumina were used for the bulk segregant analysis. So this shows the synapse, their LOD score from transcriptome analysis. And the synapse ID is on the x-axis and LOD scores in the y-axis. And this red line shows the cutoff value, post-discovery rate, 0.05. So only 10 synapses in that pedigree surpassed the cutoff line. And they had a very large effects, basically, on the disease outcome. So in other family, about 11 of them surpassed the cutoff line. So this shows the LOD values of those 10 synapses in one of the transcript was 69. And those 10 synapses you see here at this is the, for the diseased bulk. This is for the non-diseased bulk. Sorry. And then this is the random bulk here. Those synapses were completely fixed in the diseased bulks, 10 of them. But they were like intermediate in the non-disease or the random bulks. So so in addition, Eddie looked at the amino acid sequence alignment for those candidate genes, and he discovered 3 of them in family E4, and then 1 in other pedigree. So those are nucleotide binding leucine-rich repeat genes, and they were associated with synapse markers. So conclusions, in loblolly pine, we discovered that a small number of QTLs can provide effective resistance against the pathogen. Like 2 QTLs can confer complete immunity. And we provide the first evidence for the putative race non-specific broad spectrum QTLs in a conifer genome first time. And the candidate genes we discovered from the transcriptome analysis, they are similar to other plant disease-resistant genes, like NLR resistant type of genes we see in many crop species. So, based on that, research, we developed a proposal, and it was funded by NSF, NIFA Plant Biointeractions Program, to really look into the pathogen side, to understand the virulence level in the pathogen. But we use those 4 different genetic clusters, groups from the previous research as our host to see how fungal genes can overcome those host genes and what's the virulence between different single spore isolates of the fungus, virulence variation, basically. And this shows the experimental approach. So basically, we are extracting pinus spores from the gall seedlings. We know their genotypic classes, whether they have a 1 resistant allele, 2 or none. So, and then sequence the pinus spores from those pine genotypes, and then sequence it, and then look at the virulence variation in the fungal sequence data. And then one of the objectives to develop a pan-reference genome for the fungus for further research. So for this one, I'm collaborating with a plant pathologist. That's really not my background. Ralph Dean and a few others, and a core geneticists also, or pathologists working. Those are the funding resources. That's all I have.
Any questions for Fikret? I should mention that we do ask the speakers as part of this series to keep the seminars rather short, so there's plenty of time for discussion. So, yes, Steve.
What's the damage to the industry of the fungus for the rust, and then what's the status of deployment for the resistance?
So, according to some estimates, the disease damage is about 10%. So you lose 10% of the trees you plant, which may look small, but actually the economic impact is, is huge. There are some estimates talking about $100 million per year lost because of the disease. That was an estimate in 2000, probably it's more. So the deployment, those QTLs, they are in that population, just validation population. But the strategy we use to improve for disease resistance, historically, it's like we treat it like as a polygenic trait. You collect data, yes or no, calculate breeding values, and then you rank and give them ways to growth and disease. And it has been actually successful. It's a classical, selection. It works. So there are a lot of resistant families. They deploy. They get much less disease compared to others. Now we have some research going on gene stacking approach, using these two, genes from 2 pedigrees in the same clones, and developing Cas markers through genotypes and using kind of marker selection. But it only works in those pedigrees. That's the problem with outgrowth species. They can be used maybe as pollen source in a seed orchards to develop disease-resistant material for deployment. Breeding application is more limited. for those QTLs using those.
Yeah, great talk. While we're on this page of funding, what does funding look like for such a long-term process, right? Where maybe you're not doing evaluations until year 6, like you said.
Well, actually, we got pretty good results in 6 months. You know, artificial inoculation, it works very nicely. We just, you know, had that nice population already in place. It was not necessarily we developed for that research. It was developed for genomic selection and like a pilot project. And, you know, since we have pedigrees used in that population, we decided to see if it works, in, you know, if we can validate those QTLs. So there is a lot you can do with pine, even though it has a very long breeding cycles. Yeah, there are a lot of research you can do. So yeah, we convinced NIFA and NSF to give us money.
What are the mechanisms of these resistance to the QTLs? There seem to be pretty broad spectrum.
They look broad spectrum, and so I'm not sure. Are you told like a
The molecular mechanism.
molecular?
Yeah.
That's something really we don't know much about. You know, how it works at the molecular level. Maybe with that research, the new research I mentioned, that will help us to understand better. So when you work with conifers, they have huge genomes, and the reference genomes we have is very fragmented and it's almost useless. So without some really good genomic resources, it's difficult to get into some of those details. That was a challenge in ethics research.
Thank you so much for your time. It's very interesting. I have a question about the mix of pathogen you have. So, how do you know what? I'm going to explain where I come from. We work with race-specific resistance here with roses, and we know that some of our pathogens we have different races, and some of them are more aggressive than others. And so, if we were to mix all the different races, we know that some of them will overcome the other ones and will most likely infect quicker, and so the other one will be left behind. And so, how do you know that the cells that you are looking at are not just race-specific, because some of them are actually infecting only the kind that are the other one? Have you tried to look at what the spores are after the cycle is complete, just to know which race infected in which genus?
Yeah, it's a good question. Yeah, I don't know. Yes, it's interesting research question to look at, but yeah, I don't know much about it, and we didn't do any research on that part. Maybe the next Eddie Lauer graduate student can take on that challenge. Right, it's interesting. We don't know. It's really like a black box. You have a very dense inocular coming from many trees, many resources. You don't know what's infected, what's not infected.
Yeah, because we have some research that we're trying to know when we have a spot in our what races do we have? Is there more of one or the other one? So maybe with this research, you might be able to know.
Yes, that's probably should help answer some of those questions. Virulence, the variation between those pygmy spores. Pygmy spores are haplotypes. So, you know, when you get the pygmy spores, sequence information, sequence data from those pygmy spores, then you know the host genotype, and that will should help us to understand the virulence in at least in fungal population.
Yeah, do you have any idea of geographical variability or population structure in the fungus? Over a pretty big range.
Yeah, it's a big range. It's pretty much everywhere, but the virulence, disease level, severity is mostly you see in coastal plains. Like coastal plains of starting from North Carolina, South Carolina, Georgia, Alabama, Florida. And yeah, it's very wide. Mostly you see in Georgia, there are like hotspots. There are really susceptible areas. In Virginia, colder areas, it's not that issue. It's not an issue rate in the colder Virginia.
Your first two populations, they're very large vet food fields. A lot of them, 20, 30, we're only excluding 30% of variation with population. But it seems to me that should be a lot higher. It should be explaining 70, 80% of the variation. What was that for? Binary, resistant, susceptible, or is that quantitative?
It's a binary. So you get about, yeah, it's a partition in the disease incidence. Like it was 28% in the first category. And when you partition the variation into genetics and environmental components, it's about 31%, which is genetics, which is actually very high to me. But if you just look at the haplotypes, two locus haplotypes, like double resistant haplotypes in pine, they are completely immune on average. If you just average across, partition the variation into genetics and environmental interactions, 31% is genetics.
I wonder if you talk about management practices. Is there a way to maybe eliminate all alternate host folks in a 1 mile radius from the farm, or say culling infected trees very early on, or cultural practices to reduce this disease?
There is almost no cultural treatments, management practices that can help to reduce the disease, unfortunately. It's really all genetics. So yeah, once you plant them, they are left to mercy of the pathogen. Whether they are going to, in fact, if they have good genes, resistant genes, they will be resistant. One of the questions, sorry, go ahead.
No, you go.
Yeah, I frequently get that question, is there a selection pressure on the pathogen? I mentioned about the arm race between pine and the Cronacea fungus. So are we going to have a problem like planting resistant material across the south? The answer is really not really. There is no selection pressure on the fungus because think about it. There are 200 million acres of forest land in the south. It's not all pine, but so it's only, I think it's like 19% is intensively managed, maybe less. And then the managed plantations, and it's not that we plant those resistant trees everywhere. There are a lot of families people plant based on wood quality, other traits. And for fungus, there is trillions of other trees to infect. Like those are wild, unmanaged forests. So there is no selection pressure. So we are pretty safe. So yeah.
I was just going to ask, I've already betrayed my ignorance about soft pines and hard pines, so I'll just continue along the steam and betray my ignorance about conifers. But anyway, so how you mentioned you sample the megagametocytes from those seedlings.
Yeah.
How do you do that?
So when the seeds are germinated, you see like a seed coat, like a cotyledons, like green small leaves come out, and at the top of the seedling you see seed coat. So there is a tissue inside the seed, and the zygotic tissue is already germinated. So it's very easy to pull out the seed coat from the germinated seedling, and you extract that, megagametocyte from the seed inside the seed. So maybe I can find like a picture, but if you, yeah, it's very easy. I mean, just come up like, when they're small like that, it's very easy to get that tissue.
This is a general plant pathology kind of question, but at the beginning, you mentioned this evolutionary arms race, how this host pathogen relationship has been evolving for millions of years. Is there any general correlation between the length of a host pathogen interaction and the quantitative versus qualitative nature of resistance there? Or does time not make a difference on that?
Let me see if I can follow your question, whether there is a difference between qualitative and quantitative resistance.
Or if there's a tendency towards quantitative or qualitative resistance based on the length of the host pathogen interaction, right? A more recent one would be one or the other versus one that's been for millions of years would be different.
Right. So it is, at the single gene level, it pretty much follows Mendelian principles. Single pedigree, you challenge with single spore isolation fungus. But in the real population level, it's really complex. We are talking about many different of the pathogen isolates, right? Some of them are more virulent than others. And the host population is also very complex. You have a lot of trees with different genetic makeup. So the way we approach the pathosystem is not necessarily go after individual QTLs and use them for breeding. We treat it as a threshold model at the population level. There are probably many QTLs segregated in different pedigrees and many fungal isolates challenge those genes. And at the population level, we see percent disease. And that percent disease is the phenotype we see, yes or no. And we analyze as like a polygenic trait. Well, it's a binary, but we analyze using classical quantitative genetic methods, like a polygenic trait. It's a threshold model, basically. What you see on like as a phenotype, gall or no gall, actually is controlled by genetics of the host, pathogen virulence, and maybe some environmental factors. But on the surface, on the phenotype, it's a yes or no trait. So we analyze like as a height, basically, or yield. And it works. Selection works. Something we don't know how, but it's really works. If you do it over and over again.
You mentioned the tiny genome and how complicated it is, how the genomic resources aren't that great right now. Do you think that an improved genome is on the horizon, or is it still many, many years out to have a complete genome?
Yeah, we are, I think, getting there. The first reference genome of fungus-pine was published in 2014 by Dave New group from UC. But there are like 1.7 million scaffolds in the genome assembly, so it's very fragmented. So I was talking to Peter yesterday. He really gave me a great idea to do something about it. So bring the community together and then apply for the GGI to sequence the fungus-pine genome and then develop a much better reference genome. And I think that's something we're going to work on coming months.
So just to follow up on that, I think working with plant really big genomes is challenging. The resources are coming, but the nice thing, and I think your approach kind of went towards this, is that often transcriptomes stay relatively the same size, even in really, really large genomes. And so I really like your approach of doing the bulk RNA-seq analysis to kind of look for different differences in genes. I would suggest that maybe you build your transcriptomes from those segregated pools and look for that lets you look for differences in isoforms. It also lets you look at expression differences across the susceptible and resistant cultivars, not just SNPs, I guess.
No, that's a great idea. No, definitely it's a great resource. But I need help to do that. So I'm looking for a postdoc to work on that project. If anybody's interested, come to Raleigh, let me know.
Couple more questions. Steve, correct?
Yeah.
It's following the same track. I think of like corn and genome, how there's a lot of genes which necessary. So I think there's a lot of similarity in that regard with genome. Other large chunks of genome that maybe aren't present in certain sessions. Do you have any idea of that kind of genome variability?
What I know from the literature, it's really a guess how many genes are controlling those complex traits. There are estimates up to 20,000, especially for growth traits. Maybe like a good study to look at number of genes for those complex traits is the cattle. Famous genomics selection paper authors. Forgot the name of the authors. Mearson is one of them, and then two guys from Australia. They were, yeah. So they published a paper after looking at the genomic data of cattle populations, and then their initial estimates for number of genes controlling those complex traits was about 200. That was before, next generation sequencing data was available. So they revised their estimates to 10,000, like this for the milk yield, milk production. So it's still a wild guess. It would be a wild guess for pine. We don't know exactly.
I was going to say it's wonderful for us here at the Plant Breeding Center to hear a talk on forest genetics and breeding. It's something that we know is there, but we don't know too many of the details. And I'm thinking of most of the work that we do, and in a way, the day in the life of an annual, plant breeder for an annual species, and how that might be different. And I'm wondering two things. How do you pollinate pine? And second is, how do you measure these traits? Like plant height in corn, I know how to measure. How do you measure height in pine and these sorts of traits? I was wondering if you'd walk us through some of the practicalities of the logistics that are involved, just to give us a flavor of how it is like to be a tree breeder.
Tree breeder. Now, tree breeder has its own challenges for sure, and pollination is one of them. So the trees we select from those tests, we put in a breeding orchard, graft them actually. And it takes, I don't know, usually number of years they can flower. But we graft them on the top of the old trees so it can simulate flowering early. Otherwise, you have to wait 10 years. So those trees are really tall, 50 feet above the ground. The female flowers are at the tip of the tree. They make it really hard to reach. So we use those leaf trucks. So you are in a leaf truck and above ground. It looks scary sometimes when it's windy. And then we put pollen bags, isolate those female flowers before the pollen starts flying everywhere. And then, of course, we have those pollen already processed a year ago, labeled in the freezers, and then we have a crossing plan. And then we stop everything when it's time for pine to have fun. Students, staff, we go to breeding center in Georgia, and we do fish crossings in 2 weeks, and then go back home. So the measurement progeny test measurements. Yeah, so this is a typical test we measure. We measure at early age,
Okay.
like age 4 and 5. And we can measure height easier. Once they get taller, we give up on measuring the height. We don't do that anymore. It's just too much work. And actually, we collect data at age maybe up to 6, and then we abandon the test. So of course, trees, they grow for another, they can grow. I mean, the harvest age 25 years. So we think the winners we select at this age are gonna be winner at age 25. That's the assumption we make, and it's a pretty good assumption, actually. Once they, they are, winners here, they are not gonna give, they're gonna be winners. There are some long-term studies. We looked at the age genetic correlations, but pine, once they, they are dominant in the forest,